will go through a general introduction of digestion. So your digestion happens in the intestine, you all know, and intestine, and then absorption happens through this brush border epithelium, that is your mucosa. Okay, mucosal layer, and that mucosal layer, the epithelium is there. And you know the layers of your intestine, everything, histology, you know better. Okay, and then you can see here, digestion is basically nothing but breakdown. If you take something, a polysaccharide, generally you take, or everything you take is a polysaccharide in your diet. For example, you take protein, it is a polysaccharide. You take starch, it is a polysaccharide of glucose. You take lipid is compound molecule, two or more than two molecules are attached to form lipid. So this all has to be broken down into simpler form in order for them to get absorbed. So they will be broken down into simpler form. This is called digestion. And after this simple form, it will reach, this is simple form, or you can say monomer form, monomer. So it will reach your cell. Inside cell, it will be digested, it will be further broken down by catabolism, or it can reassemble to form your part of cell. So there are two processes. One is catabolism and further breakdown, or second process you will see anabolism. We have been talking about this catabolism and anabolism. Generally anabolism, not strictly but generally, anabolism corresponds to fed condition and catabolism corresponds to fasting condition and starvation, fasting and starvation condition. So this is general rule. Okay, so catabolism and anabolism based on requirement of the cell. So we have seen, we are going to talk about digestion. So membrane, you can see this is, so this various product will come, it will be digested and it will be absorbed like that. And there can be intracellular digestion in by lysosome. So you can see luminal digestion is, there are two types of digestion. You can see luminal digestion, and we will talk about that. Membrane has many enzymes. Brush border enzymes are there. There are many brush border enzymes, all for fat digestion, not for fat digestion, for protein and for carbohydrate digestion. You will see luminal enzymes are there. That enzymes are attached to the membrane of epithelium. So this is called membrane digestion. Another is luminal digestion. So enzymes that are secreted in the lumen of intestine, okay, they will digest they will do the digestion inside the lumen. This is called luminal digestion. So this pancreatic enzyme does luminal digestion, and generally intestinal enzymes are attached at the brush border. They will do membrane digestion. And intracellular digestion is something you will see the lysosome. You will see generally lysosome, they will digest. So three type of digestion you will see. Okay, membrane digestion, luminal digestion, and intracellular digestion. So these are the enzymes of digestion that we see generally. For carbohydrate, we will see amylase. Everywhere you will see amylase. For carbohydrate, amylase is the main enzyme. For protein, there are many enzymes for protein digestion. You can see from pepsin, then trypsin, chymotrypsin, aminopeptidase, and carboxypeptidase. Many enzymes you will see. For fat digestion, you will see only one enzyme, lipase, and there will be co-lipase helping this lipase. So lipase and co-lipase will be there. So co-lipase will be helping lipase, and there are phospholipases. Again, lipase, acting on phospholipid is called phospholipases. So there are 3 types of enzymes will be there. All will have name at last lipase. So lipase is the enzyme to digest this, and amylase is for digestion of carbohydrate. And for this nucleic acid, we have already done this. I told you the nucleases, DNAs, RNAs will digest this DNA to RNA, and then nucleotidases is there, nucleosidases are there, and then catabolism. We have done this. So the digestion is done, but we will not talk about this nucleotide digestion. We will talk about this carbohydrate, protein, and lipid digestion. So protein has, you can see, 4 enzymes. We will talk in detail about this all enzymes. Now let's talk about, today we will talk about carbohydrate digestion. So this amylase, this is pancreatic amylase, this is luminal digestion, and then disaccharidase will carry out membranous digestion. Okay, so we will talk about luminal and membranous digestion. So there are two. So product of exocrine part of pancreas is very, very important for digestion, and this product of exocrine part of pancreas has enzymes and electrolytes. So enzymes, you can see enzymes are protein digesting, fat digesting, and carbohydrate digesting enzymes. So proteolytic enzymes are, these are the proteolytic enzymes. You can see these are lipid digesting enzymes. I told you all has lipase at the center, at the end. Only here is one esterase. Anyway, lipases attack on ester bond because all fat has ester bond. So that is how you can see here cholesterol esterase and pancreatic amylase and this ribonucleotidase and deoxyribonucleases, ribonucleases and deoxyribo. These are the enzymes that are present in the pancreatic juices, and these are the products. So chloride and bicarbonate, these are the actually part of the pancreatic juice. Main part of pancreatic juice is bicarbonate and chloride, and very important part of this pancreatic juice. So chloride is secreted by different cell. You can see, you already know, I think you might be knowing from your histology class. So acinar cell, chloride, and then you can say ductular cell, bicarbonate, and you will know this through this image, So I told you what acinar cell and ductular cell. So you can see secretion of chloride here and here. From other cells, secretion of chloride. So though this seems very complicated picture, it is the main purpose of this picture is to show the action of certain things, like parasympathetic nerve will act on M3, so that you will remember. And then this is cholecystokinin. Cholecystokinin will act on its own receptor, and after binding, cholecystokinin is what it is responsible for secretions of what? Enzymes. And you can see parasympathetic response, they both will secrete what? They will cause secretion of enzymes here. Okay, enzymes here. And you can see here this secretin will cause secretion of chloride and bicarbonate. Chloride and bicarbonate. That is what I want to show from this picture. And you know this. After digestion of this through this enzyme shown by scissor. So this after digestion, after it is being absorbed, some part of this carbohydrate will reach here also, pancreas. In pancreas, beta cell is there. So this beta cell, there inside this is islet cells, has beta cells, and this beta cells actually will be stimulated by glucose. Okay, beta cell will be stimulated by glucose, and this actually insulin. So the beta cell will be stimulated, yes, glucose, and glucose will release insulin. Okay, glucose will cause release of insulin, and this insulin again, you know, induce the metabolism of glucose. So this glucose digestion, and then after absorption, this insulin. So this pancreas is performing in digestion also, digestion by secreting this digestive enzymes, and at the same time by releasing insulin and glucagon from islets of Langerhans and cells, alpha beta cell insulin and alpha cell. After secreting, this carbohydrate digestion is also carried out by this pancreas, and metabolism, both digestion plus metabolism, both is carried out by pancreas. Remember, correlate this with when pancreas is not there. If you have to understand something, you'll have to understand this. When pancreas is not there in condition of acute pancreatitis, when pancreas is not functional, acute or chronic pancreatitis, so what will happen? Your digestion will affect, and at the same time, after with digestion, your metabolism of carbohydrate, protein, and fat will also be affected. digestion of your food and metabolism of your food will be affected. That is why pancreas is very essential for your survival. If pancreas is not there, people will not survive, because both digestion and absorption, digestion and metabolism both are affected. Okay, so this is the role of pancreas. So you know this. So cholecystokinin for synthesis of enzymes, and then secretin is for synthesis of this, to secrete, secretin is to secrete bicarbonate and chloride, you can see chloride. So this is the function, and both will act through, all are G protein coupled receptor. This is also G protein coupled receptor, this is also G protein coupled receptor, and secretin is also a G protein receptor. All are G protein. You can see CAMP here. Here you will see something else. You will see calcium, IP3 DAG pathway. You will see something else here. So IP3 you will see, IP3 DAG you will see through this. This act through this muscarinic receptor act through IP3 DAG pathway. Okay, so the mechanism will be a little bit different. The G protein will be different, but all are G protein coupled receptor. This is how secretion of enzymes, all enzymes, and this fluid will happen, and it will reach pancreatic juice. It will form pancreatic juice, and pancreatic juice come out of this. So you know CFTR, okay? CFTR. So this chloride is secreted through this CFTR. Chloride form. Chloride secretion occurs through this CFTR. This is cystic fibrosis transmembrane conductance receptor. You know this CFTR receptor, if there is defect, it will cause cystic fibrosis. And in that condition, pancreas is affected. Your pancreas will have problem. Okay, and that is why when pancreas does not, it is not there viable. If pancreas is not viable, it will cause problem to the patient. So survival is not possible without pancreas. This is the one of the condition that will cause damage to pancreas, because it will affect this. You have studied this in detail in genetics. So absorption of nutrient. So this is the very, very important slide. And this is very important. Why? Because you will, in one slide, you will know the absorption, the site of absorption of important constituent. And if you know this simple slide, later you will not have problem in understanding absorption, digestion and absorption. So digestion of carbohydrate, actually absorption of carbohydrate will happen here only. So it will not go beyond. It will happen in duodenum and jejunum only, because digestion of carbohydrate is easy. And that is why digested immediately, very fast, and absorbed very fast. And that is why this carbohydrate will not reach beyond this. So it will, the digestion and absorption of carbohydrate happens within duodenum and jejunum. It will, all carbohydrate will be absorbed there. But the digestion and absorption of this fat and protein is little complicated than carbohydrate. That is why it will reach up to ileum, terminal part of ileum. So absorption happens till the ileum. Whole intestine will absorb fat and protein, whole intestine. So carbohydrate is required duodenum, jejunum only, and this require whole intestine. These two are macronutrient, and these two micronutrient are very important. Okay, so these two are iron and calcium absorption happens in duodenum. Iron and calcium absorption happens in duodenum. So I will tell you, there is 3D formula for calcium. So what is 1D? 1D is duodenum, second is DCT. Absorption happens for calcium, absorption happens in duodenum. Then whatever calcium is going to excrete from the urine, it is again be reabsorbed in distal convoluted tubule. So second D, first D is duodenum, second D is DCT, and third D is, it has absorption of this calcium is requires vitamin D. So that is why you have to remember 3D for calcium: DCT, duodenum, and vitamin D. Influence calcium metabolism. And iron also will be absorbed from duodenum. So with calcium, you can remember iron also. Iron will also be absorbed from duodenum in divalent form. Iron always absorbed in divalent form. It is converted into divalent form from this trivalent form, and then it will be absorbed. And here you can see calcium is already a divalent. So if whatever you see absorbed, whatever molecule you see, iron you see absorbing from the intestine, it will be absorbed in divalent form. This is a universal thing you will see, and not with sodium. Sodium is already a monovalent, but whatever you see for copper, for iron, for zinc, so whatever element you see macronutrients, they generally they all absorb in the form of divalent. They all absorbed in divalent, and iron is not an exception. Okay. So these are the, and this is very important. The most important here is, one of the most important is vitamin B12 is absorbed in terminal ileum. Okay, vitamin B12 is absorbed in terminal ileum, and bile salt is also absorbed in terminal BB. You can remember BB.